I would like to begin by acknowledging my co-authors. They're the laboring oars in this effort, and I'm only up here to obfuscate and misinterpret the results. But they did the hard work on this, and I want to make sure they get the credit. Some of the slides that you're about to see are sort of repeats from what Marty just said. He handled the terrestrial vertebrate side of things, and we handle the aquatics. So there's going to be a bit of overlap here, but our study began in the late 1990s with this retrospective space for time substitution type study of a number of sites along Washington's Olympic Peninsula. Basically what we found for these streams, about 2/3 of them of which were fish-bearing streams, was nothing that we really hadn't already learned before from a variety of studies. First of all, we tended to find fewer fish in late seral forests and more fish in very young forests or forests that had been harvested within the last 10 years. But that fish abundance tended to be correlated primarily with habitat features at the site, including light gaps, organic matter inputs, nutrients, and so forth. And again, this was something that we probably known for 30 to 40 years, so we didn't find much new there. And on the amphibian side, we tended to find the reverse situation where we found more in-stream amphibians, although this is the terrestrial phase of the giant salamander. We found more salamander larvae in late seral forests, and there was a negative correlation between salamander abundance in the streams and road density, which we interpret to mean that somehow their abundance was related to sediment inputs. But be that as it may, this was a correlation type study and didn't really give us the insights that we hoped we would get at the outset. So we decided to move on to a second phase of the REM study, which would utilize these adaptive management areas that originated from the Northwest Forest Plan. These are places that were set aside to provide flexibility and to test many of the management actions and scientific assumptions that went into the Northwest Forest Plan. And that led to a series of surprises, and you'll see several slides in my presentation that talk about the surprises that we encountered in setting up the study and then in executing it. And the first surprise was that despite our good intentions when we approached several national forests in western Washington to implement this experimental whole watershed treatment approach, which would have included sites with Northwest Forest Plan buffers, we were told that, yeah, it's a good idea, but we can't, our planning rules and our other considerations at the forest level would not allow us to run the kinds of studies that we wanted to do. So we turned to another partner, in this case the Washington Department of Natural Resources, as Marty described, and our focus shifted from down to southwest Washington in the Willapa Hills, where we put in a before-after control impact study of alternative riparian treatments in small watersheds. A couple of things. One is Northwest Forest Plan buffers were not on the table here. These are state-managed lands. And second, none of our treatments involved thinning, and I realize that this conference primarily is about thinning. But here our goal was to test the riparian reserves in the smallest of the small, the streams that Bob talked about and this morning. And this is a shot of the post-treatment of a post-treatment hillside and one of these clusters of small watersheds that included unlogged controls, these so-called continuous or fixed width buffers, the patch cut buffers, and the no buffer treatments, which is on one side or another. And this is where they were located. And if you look at several of these watershed clusters, you'll notice that some of the clusters, as Marty pointed out, did not include all of the treatments, and at least two of the clusters included duplicate treatments. The split roost site had 2 clear cuts. The tag site in Willapa Hills, or the Capitol Forest site, had a couple of patch treatments. So it wasn't a balanced experimental design, although that was what we wanted, that's not what we got. Our second surprise was that even though we had good intentions to try to do this properly from a statistical standpoint, not every treatment was implemented in every block of watersheds, and the controls were always located at one end or the other of a cluster because of road considerations. The third one was that despite our best intentions, the treatments didn't go as planned. As Marty said, in some places the timber sales took place a year sooner than we anticipated, and we had, instead of the treatments all taking place after the second year pre-treatment study, some of them took place after the first year. And in another case, in a few other cases, the loggers themselves didn't always follow the guidelines with regard to the buffer edges. Nevertheless, here's what they looked like. This is a picture inside out from a control watershed, a fixed width buffer watershed, looking downstream through one of the patches. And this is a photograph of one of the sites that was clear cut. Now in Washington, they're so-called type 5 streams, which is equivalent to what are here in Oregon type N streams. There is an heavy equipment exclusion zone next to the stream, so that you don't have tractors and things going through the channels, and they're not supposed to use streams as yarding corridors. But nevertheless, you can see that there is a fair amount of slash entry into some of the sites. So they were basically harvested right down to the channel's edge. The fourth surprise was that we really hadn't figured on variability in weather, and that was something that hit us, at least on the aquatic side, fairly hard. From 2001 to 2004, we experienced a drier period of years than normal. Basically, they were sort of drought conditions that affected our ability to sample, and it turns out that beginning in 2004 for the next couple of years, that was our post-treatment thing. So we sort of had a weather shift that took place right about the time that the treatments were implemented, or shortly afterwards. Another surprise was that we had some post-logging site preparation treatments, in particular spraying herbicide on some of our sites afterwards, that we hadn't figured on, and they didn't tell us this was going to happen. And so when you're doing things like studying riparian invertebrates and aquatic organisms in the streams, it's always nice to know something like this is going to happen. We didn't find out until after they had happened. So the sixth surprise was, and Marty showed a photograph of this, that in a couple of the post-treatment years, we had some fairly significant windstorms that basically caused some of the treatments to sort of mutate into other treatments. This is a fixed width buffer that now is starting to look a lot more like a patch buffer during the thing. So, and along with the wind throw aspect of it, there was some sedimentation in the channels associated with upturned root wads and the like. Okay, so now I'm going to go through the results. I have to say that each of the team members have analyzed their data in a somewhat different way. I'm not going to spend a lot of time focusing on the method of analysis, but try to take you basically to the conclusions. This was the first year post-treatment temperature effects, and if I may direct to your, maybe this one doesn't work. Does this have a pointer?
Yeah, there's narrow band.
Oh, got it. Okay. All right. I don't know if you can see it. There it is. The first year post-harvest sites, the average temperature increase during the summertime in the clear-cut catchments was 1.5 degrees. We expected the continuous buffer and the patch-cut buffers to be intermediate, and in fact they were, but one would normally think that the continuous buffers would experience less of a temperature rise, the patch-cut buffers to experience more of a temperature rise. That turned out to be not the case. It was the opposite of that, and there's a reason for that I'll talk about in a moment, but basically that we did see a temperature increase in the sites, and this has been published in a paper by Jack Janisch, Steve Wanzel, and Bill Anger recently. In effect, what we found were that the temperature increases in these headwater streams were small. The trend did not always match the buffers design, and the responses within treatments were highly variable. We were surprised at how much temperature variability there were among streams that were located right next to each other on the same hillside, and yet they were, one stream would be warmer than the other consistently, and there were, it turns out that there appeared to be pretty good reasons for that. One had to do with surface area exposure and the amount of hypereik exchange. If the stream spent more of its time, and again, these are sort of permanently intermittent streams, if the stream spent more of its time on the surface, it tended to warm up faster. If the stream was underlain by coarser substrate, there was a lot more hypereik exchange, and the water temperature appeared to be somewhat cooler. So that was a complicating factor here. I'm going to go through what we found for some of the invertebrates and begin with the terrestrial ones. These were the most common terrestrials that we had that were drifting in the streams themselves. These were mites and springtails, and the results were that these two groups increased in the clear-cut streams compared to the control and fixed-width buffer streams. And I should mention that the patch-cut buffers are not included in the bug analysis because of small sample size and high variability things. So really, we're only looking at three conditions here: control, a continuous or fixed width buffer, and a clear cut system. Anyway, terrestrials increased in the clear cuts in the summer, and they also increased in the clear cuts relative to the control streams in the fall. In the spring, there weren't many differences among the sites. We didn't sample in the winter because basically we didn't have access to a lot of the sites because of the snow and the roads were closed. On the other hand, the aquatic invertebrates showed just the opposite pattern. Mayflies decreased in the clear-cut streams compared to the control streams in the fall. In the spring and summer, the streams were not significantly different. Midges, one of the most common and ubiquitous a group of organisms in small streams, decreased in the clear cuts relative to the control and fixed width streams in the fall, just as the mayflies did. And in the spring and summer, again, they were not different from one another. And so if you then sort of slice and dice the bug data in a different way and group them into various trophic functions, those that scrape algae, those that shred leaves, and those that collect and gather detritus, and forget about the traditional taxonomic definitions, we find that the scrapers decreased in the clear-cut sites relative to control and fixed in the fall. You think there's a pattern here. The collector-gatherers decreased in the clear cuts compared to control and fixed in the fall. And in the spring and summer, not much difference. So actually, the results were fairly consistent. Now, we did look at one more group that I want to report on here, and those were riparian mollusks. You may have heard on the news yesterday that the Fish and Wildlife Service is considering slugs and snails now as potential candidates for listing, and so maybe these data are more relevant than they might seem. What we found here was much more confusing. This is simply pre and post comparisons in the different groups of treatments, clusters of treatments. But basically what we found was that there were fewer riparian mollusks, and these are very small snails, very small slugs, and they were lower in the capital forest block and in the other blocks even before the treatments began. We don't know why. Maybe this was just happenstance, but the fact that they tended to be lower in the clear-cut sites carried over right on through the treatments to the post-treatment period. Again, we don't know why, but we did notice that the differences between treatments, again with the clear cuts being the lowest and the others being higher, was greatest in the fall. We attribute a lot of the differences here to a fall down in the input of terrestrial organic material after harvest in the clear-cut sites. We have no other way of explaining the consistent differences we saw in the aquatic invertebrate communities other than to say that we didn't see a boost in aquatic primary production that we thought we would see. In fact, what we saw was a fall down in most of the taxa that probably was related less to what was going on in the algal and in the periphyton community and more to what was going on in the community specialized to process allochthonous organic matter. And in fact, we did see quite a fall down in terrestrial organic inputs in the clear-cut site. So, to conclude, it was very difficult for us to set up a nice, clean, balanced BACI type study. This was a lot harder than what we thought. There were lots of implementation problems, and the control sites weren't true statistical controls in the sense that they were often different than their adjacent treatment partners just because of natural variation. Second, we didn't follow. I mean, really, we didn't have any idea that the variability was going to be as high as it was. And our plan to do a 5 to 6 year study with 2 years of pre-treatment and 2 to 3 years of post-treatment was really inadequate for establishing a response among many of the metrics that we were going to try to do to follow the results. The third was that, boy, were we surprised. All along the way, we were surprised by both natural and anthropogenic changes that we hadn't anticipated. And finally, and this is sort of the punchline, the biological punchline, is that that we did see, even with all the shortcomings in the study, we did see a change in the biophysical conditions in the headwater streams. In the sites that had buffers, the changes were quite minor. In the sites that were clear-cut, the changes were statistically measurable, but not huge. I mean, the communities just didn't collapse. We still had them there, but you could see a difference after logging. And that difference was primarily expressed in the fall. That was a bit of a surprise. We thought spring and summer would be the critical time, but I think our results are more consistent with what Jason talked about this morning. So, in terms of lessons learned, we would suggest to other scientists to think of watershed experiments as interventions. No single design, including BACI, is not going to be best suited to answer all of the management questions. We probably should have spent more time characterizing pre-treatment variability, but we didn't know. If we had it to do again, we'd allocate longer time to trying to understand that. If you're going to implement an experiment, stick with it, warts and all, right through the end. You can still learn things even with all the surprises. Don't be tempted to switch treatments in the middle of a study. If you're going to dedicate one area to one treatment, stay with it. And finally, don't be afraid to investigate novel response metrics. An example that I'm going to give is millipedes. I didn't talk about millipedes here because they didn't show a response to logging. But in the process of studying the millipede communities in the riparian zones, we came up with something like 3 new species and a new genus of millipedes that were new to science. And although managers may roll their eyes and shrug their shoulders, in the long term, science is best served by better understanding the communities that we have there. So I think that part was worthwhile. So finally, I'd like to thank our cooperators on this. We've had a number. This is a aerial shot of the Capitol Forest site with Google Earth, and I'll be happy to answer any questions if there's time. One question.